Good morning, ladies and gentlemen, present at BrainConnects 2019. My name is Eric Ho, and I am a faculty member with the Department of Electrical and Electronics Engineering of University Technology Petronas in Malaysia. I wish to express my heartfelt thanks to Dr. Jacqueline Dominguez and her team for graciously organizing this BrainConnects 2019. And despite their very best efforts, I regret being unable to join all of you this morning due to unforeseen personal and family illness. I also want to express my long-standing gratitude to Professor Toshiharu Nakai for organizing yet another edition of BrainConnects, which is such a useful platform to bring together like-minded scientists to exchange ideas and discuss collaborations. This morning, I wish to share some thoughts with you on how to perform neuroimaging analysis to ascertain differences in brain volume and neurofiber tracts, which has become so useful to describe the changes in the brain occurring as a result of neurodevelopment, neurodegeneration, or onset of neurodiseases. I will also briefly walk through some instructions on how to use publicly available software to perform these analyses. Conventionally, radiologists and sometimes neurologists perform visual assessment on MRI images such as this by inspecting axial coronal or sagittal slices. The accuracy of assessments, which is good enough for diagnosis and something that is not easily replicated by computers, depends largely on their background, their training, and their expertise and their experience. About 2 decades ago, computational scientists began asking the questions: what more can we achieve with using the increasing computational power that cannot be done with visual inspection? And so they thought about it and found that if computational tools were able to measure and estimate subtle differences in the structure, such as the volume, the shape, or the thickness of local brain regions, this would be a new, interesting, and complementary analysis that could not be done by visual inspection alone. And because this would be algorithmically performed, there would be an objective and repeatable and fair way to make comparisons. So this is the origin of the shift from qualitative to quantitative analysis. So what have we learned from the preliminary studies of structural differences? Early studies have established a link between cortical thickness, gray and white matter volume, with IQ. So the figures on the left side of the slide show the brain regions that were most sensitive, whose cortical thickness and volume were most sensitive to differences in intelligence. A more recent study in 2015 also established that the volume of these specific brain differences were highly correlated and linked to functional activity across 15,800 subjects spanning 193 studies of 6 major psychiatric diseases, which includes schizophrenia, depression, addiction, and anxiety. Besides the links to cognition and disease pathology, scientists have also found that the structural features such as volume and thickness have a heritable component, that is, they are genetically linked. The image shown here shows the brain regions, the degree to which the brain regions are linked to genetic factors, where those regions colored more to the red tend to have a higher dependency on genetic factors. And using these facts, the Enigma Consortium, which is a global grouping of neuroscience labs who pool data worldwide, have started investigating how structural features could be biomarkers of genetic phenotypes linked to various diseases. So such structural volume and thickness studies have been shown to be useful for both cognitive performance, disease pathology, as well as neuroimaging genetic studies. How does one then perform such analysis? Structural brain differences are computed via a class of algorithms known as voxel-based morphometry. As all of you are well aware, MRI images represent the three-dimensional brain as a stack of cubes, illustrated here, where each cube actually has an intensity value, which hints whether it is a gray matter tissue or white matter tissue or elsewhere. Voxel-based morphometry can be performed with two approaches. The first approach, called volume-based segmentation, tries to identify voxels that are associated with only gray matter or only white matter. And it tries to also map the location of individual voxels to known brain regions. So in order to estimate the volume, the tissue, the gray matter or white matter tissue volume in a particular brain region, the software just counts the number of cubes that have been identified in that region. Now, of course, this analysis method is prone to an effect called partial volume effects, where maybe the brain region actually does not, the boundary of the brain regions actually lies in the middle of this sphere. The second approach to assessing structural brain parameters is to try to fit a surface, a sheet, onto the upper boundary and lower boundary of the gray matter tissue layer as well as the white matter tissue layer. Once the mathematical representation of these surfaces is obtained, it is a trivial matter to obtain the thickness, which is the spacing, the distance between these two surfaces. So voxel-based morphometry software that I will describe will perform this analysis either using the volume-based approach, that is to get tissue volume measurements, or the surface-based approach, which is to get thickness measurements. In order to make statistical comparisons between multiple subjects for specific regions, each of the individual brains has to be registered to a common coordinate system. This common coordinate system is known as a template or atlas or reference brain. Now on to the software. I will cover option 1, which is a collection of MATLAB-based software called MATLAB, SPM, and the Computational Anatomy Toolbox, which you can download and install using instructions from this link. I've chosen this example because it has a wonderful graphical interface, and it is can be run on multiple platforms, Mac, OS, and Windows. Alternatively, if you wish to explore a more powerful approach, feel free to explore the free surface software found at this site. To start the software, enter this command line in MATLAB, and this green window will emerge. Click on the toolbox button here and select CAT 12, and this gray window will emerge. These are the two main windows which form the interface of the software. There are 4 general steps in the voxel-based morphometry process. The first step is to convert your MRI image files into a commonly used format called NIfTY, which is accepted by all publicly available neuroimaging analysis software. The next step is to register, segment, and estimate the volume and cortical thickness parameters, followed by a smoothing step, and finally the statistical analysis step. For the first step, go to the green window and click on this button, which will automatically perform the conversion for you. This software accepts both T1 and T2 anatomical MRI images from a variety of MRI sequences such as FSPGR and PRAGE, and can accept images from both a 1.5T and 3T scanners. It is highly recommended that your MRI images have isotropic resolution with at least 1 millimeter voxel size in each dimension. The next step can be achieved by clicking on this button labeled segment data on this gray window. Clicking this button achieves a flurry of steps, which is the main computational workload of the software. The first class of preprocessing step seeks to correct the any inhomogeneities or in the acquisition of the images. These typically refers to bias field inhomogeneities that are found in 3T images, T1 images, and this appears as a gradual change in intensity over the image. After bias field correction, the software attempts to register, that is to deform individual brains according to a specific template so that all the brains can be compared with reference to a common coordinate system. In the case of the CAD software, the template is derived from the IXI dataset, which has been derived from 555 healthy control images, which have been registered to the ICBM MNI 152 template. The last step is actually to remove the skull tissue to retain only the soft tissue of the brain. Subsequently, the software attempts to label each individual soft tissue voxel as either belonging to gray matter, white matter, or cerebrospinal fluid, which are also the ventricles. And once the gray-white matter delineation is performed, the software will proceed to estimate the local volume and thickness of each individual voxel according to the normalized template brain. Once this step has been completed, go back to the green window and click on the smoothing button. The smoothing is actually a local averaging, that is spatially local averaging of the cortical parameter estimates, that is averaging of the local volume, of the local thickness, using a Gaussian spatial filter. When you can specify the width, it is recommended to use a full width half max of 8 millimeters. The reason we perform smoothing is to increase the statistical power of the test. Smoothing is equivalent to reducing the spatial degrees of freedom. The final step is actually to fit an explanatory model of our cortical thickness and volume estimates in terms of our desired parameters such as age, cognitive score, total intracranial volume. So the independent variables or the explanatory variables are called X. If they are categorical, we refer to them as factors. If they are numerical, meaning they are a number, they're a fractional number, we refer to them as covariates. This step, we are trying to ask the question, can our cortical thickness or local volume be predicted from parameters such as our independent parameters, such as our factors and our covariates? And we try to the software tries to estimate the beta values here, which allow us to predict the value of the structural brain parameters from our factors and covariates. It is recommended for structural brain imaging to always include the total intracranial volume as one of the covariates, and this is automatically computed by the CAD software using this button here. Once you have computed a model, an explanatory model, a model that could explain your structural parameters in terms of the correlate and covariates and factors, we want to perform a hypothesis test to see if this explanatory or predictive model is statistically significant or not. The simplest way is to specify a contrast matrix, that is a row matrix of all zeros, with a non-zero with a unity value only at that particular covariate or factor in which you want to test. Setting this contrast matrix is equivalent to performing a one-sided t-test with this null hypothesis. Alternatively, you may also perform an f-test, which is equivalent to performing an ANOVA, but an ANOVA is now performed on the model parameters, that is the beta values, rather than the measured parameters, which is the thickness and cortical estimates. This is one type of statistical analysis that you can perform. The next type of statistical analysis is to compare the significance between two cohorts. In this case, we actually have the two different factors as independent factors. That is, if group A is one class, one factor, group B is one factor, and this type of design matrix, which we will input into the software, is actually trying to build first two different statistical models and then performing a statistical test on the difference between the beta values of the two groups. It is possible to perform hypothesis testing between more than two cohorts by adding more groups and building the contrast matrix appropriately. In order to perform this model building test step in the software, go to this gray window and click on this button called Basic Models. You can choose the T or F test by clicking on the design line here, and you can enter the covariates and factors and their values by clicking on this line here. To actually perform the statistical test, go back to the green window and click on Estimate. And after the statistical analysis is completed, you may view the results by clicking on the Results button. This would conclude the voxel-based morphometry analysis process.